All right, I want to talk about meiosis. Meiosis is involved in sexual reproduction because sexual reproduction involves the fertilization of egg and sperm. And so, in order to counteract the combination of the chromosomes from egg and sperm during fertilization, meiosis has to cut the number of chromosomes in half for the egg and the sperm. So meiosis is going to be divided into two cycles of the same four phases from mitosis. You have prophase, metaphase, anaphase, telophase. But the difference is during meiosis 1, you have prophase 1, metaphase 1, anaphase 1, telophase 1. And then during meiosis 2, you have prophase 2, metaphase 2, anaphase 2, telophase 2. So ultimately, you're going to start with one diploid cell and you're going to end up with four haploid cells. So I want to go through especially meiosis 1, because that's where most of the differences happen. So prophase 1 is where the majority of the differences are between meiosis and mitosis. The first thing is synapsis. This is when you have two homologous chromosomes that line up to each other, and they actually kind of, you can see here, they form a tetrad. Tetra meaning four. It's a set of four, so you got four chromatids there. And it is possible that during synapsis, crossing over may occur. Now, crossing over is when chromosomes exchange genetic information. And the important thing about this is that it doesn't happen every single time. It doesn't happen the same way every time with the same chromosomes every time. So, literally, every single time this happens, you are getting a different genetic recombination in your chromosomes. This is extremely important for genetic variety, which I talked about in class, it feels like a year ago, when I was talking about the pros and cons of asexual versus sexual reproduction. Sexual reproduction has a disadvantage in the fact that, one, you need a mate. You can't just mate with yourself. You need to have another genetically different individual to mate with. And two, it takes a lot longer. But the huge advantage, which makes all the difference, is that you get genetic variation, which allows species like human beings, when you have a global pandemic like COVID-19, that prevents all of us from dying all at once because we are genetically different. We are able to survive different viruses and different bacteria, different infections differently because we all have different combinations of our genes. So that is the biggest difference between prophase 1 of meiosis 1 and prophase of mitosis. You have synapsis, where they line up and they exchange genetic information. Now, the second difference is during metaphase 1. If you can remember, metaphase in mitosis is where the chromosomes line up in the center. Pretty easy and straightforward. But the very important part for metaphase 1 of meiosis is that lining up is random. And the reason why that makes a difference, if I can share another screen. Wait for it. Okay, so here I've got some four chromosomes, and let's say we have metaphase 1. So this time, this chromosome is going to line up here, this chromosome is going to line up here, and this chromosome will line up here. Now, the important thing, what happened to half my chromosome? Oh, damn. Wait a minute. Why? Why? Okay, very strange. The reason why that makes a difference, I'm gonna just try to talk my way out of this, is because, let's just say that's a chromosome, okay? Let's say they line up this way, and during anaphase, in mitosis, remember that was when the two chromatids were separated. So if this were mitosis, this dude would go over here, and this dude would go over here, right? And you would do that for all of your chromosomes. But this is not mitosis, this is meiosis. So during anaphase 1 of meiosis, the entire chromosome is going to be separated. So these two go over here, this mutant chromosome goes over here, and this one goes over here. Let's see if I can clean that up a bit. And what would it be like that maybe? Okay, so that's, let's say, for this particular round of meiosis. But again, like I said, lining up is random. So next time, maybe it's like this. They line up like this. So during the second, not meiosis 2, but during a different meiosis rendition, you have this guy going here and this guy going here. Hopefully these are different than the last combination I did. And this one goes over here, right? Does that make sense? Or let's say now you've got, let's take this one here. And the third time they line up like this. And so now you've got this chromosome here and this chromosome here. I'll just do those together. And these two go over here. So you see, every single time you get a different combination. That's the difference. It's also important to remember that now you've got two new daughter cells, which are now haploid, because they have half the number of chromosomes. Even though they contain 2 copies of each chromosome, you do not have, if you understand my meaning, you do not have this big giant chromosome on this side. It's missing. Hopefully that makes sense. Now let me go back to neat share. Okay, so that is why metaphase 1 is really important, because lining up is random. Now you still have the kinetochore fibers. Remember, kinet means to move, so kinetochore fibers are going to attach to the entire chromosome. And during anaphase 1, which you can see here on the left, they're going to pull the entire chromosome, consisting of 2 chromatids, to opposite ends of the cell. This is part of something called independent assortment, because separation is random. Random chromosomes are grabbed and pulled to opposite ends of the cell. So lining up is random and separation is random. These all lead to genetic variation, because you get this mix up of maternal from your mom and paternal from your dad chromosomes. And then you have telophase 1, which is pretty straightforward. You have the cleavage furrow that develops in the middle, and you get 2 new offspring or daughter cells. Keeping in mind again that each new cell is only haploid, because we have divided the chromosomes in half. Right. But the important thing to remember is that you still have copied each chromosome. So even though they are haploid, there are still 2 copies in each cell. Now, meiosis 2, you do not have the S phase, because meiosis 2 happens right after meiosis 1. And so the stages of meiosis 2 are much more like the stages of mitosis. I'm just trying to go back to, go back to my iPad. Can I share that screen? Okay, so let me move these more in the middle so I got some room. I'll go over there. All right, so now during meiosis 2, you're going to have this dude separate, this dude separate, this dude goes over here, and this dude. This is more, like I said, more like, these are random things in here. What, I don't word, oh, there. Using a new app, sorry. What the hell? Okay, I don't know what I did, but basically I should only have 2 blue lines. No, I should have 4. Oh my God, I am losing my mind. All right, I'm just gonna make it like that. Okay, so now you've got, oh, what the hell am I doing now? All right, I think, if I am correct, and I remember that I did this right. So this should be 1 daughter cell, 3 and 4. Hopefully you guys paid attention, because I think I screwed that up, but you get my meaning, hopefully. So that is meiosis 2. No, go back here, new share, go back here. Come on. I don't know how people do this for a living. Oh my God. Right, so just to recap, you have meiosis 1. During prophase 1, you have synapsis, where the 2 homologous chromosomes line up next to each other. Remember, the homologous chromosomes contain the same genes, not necessarily the same version of each gene. For example, for this chromosome, maybe this has eye color on it. And so they'll both exchange different versions of eye color, is what I'm talking about. If you have crossing over occurring, it occurs randomly on random chromosomes in random times, and so that results in genetic recombination. Metaphase 1, lining up of the chromosomes randomly at the metaphase plate, and you have random independent assortment of the entire chromosome to opposite ends of the cell, which of course means that your new cells are haploid. And then you have meiosis 2, where you have prophase 2, where the centrioles and the centrosomes form, the mitotic spindle forms. Metaphase 2, where the chromosomes line up in the middle. Anaphase 2, where the chromatids are separated. And then finally, telophase 2 and cytokinesis 2, where you end up with 4 haploid cells.